Hey guys, it's Lance Speaker here. Yes, that's my name. It's not something else, like an actual Northwood student's name or something else. But anyways, hey guys, it's Lance here, Lance Speaker. And today I'm going to be reading The Genetic Molecule, something we've all been assigned in science class, be it from Zaleski or somebody else. I don't know who your teacher is, to be honest. And you know that my teacher's Zaleski now. Whoops. Anyways, I hope you enjoy the reading. It's a little bit slower at first, but if you like faster paced readings, it gets a little bit faster in the end. Or if you're using, well, what else would you be using? In YouTube, there's an option which you can speed up my speaking. So if this is a little slow for your taste, just hit the times 2 button and then get done in half the time. Yeah. Anyways, hope you enjoy, guys. Within each cell, there is a single molecule, deoxyribonucleic acid, or DNA, which controls both cellular activities and heredity. Since the discovery of nucleic acid over 100 years ago, many hypotheses regarding the structure and function of the DNA molecule were debated. Significant experiments, testing, and some technological advances were required to achieve our current level of understanding of this critically important molecule. Miescher's early research. In 1869, Swiss biologist Friedrich Miescher, I hope I'm pronouncing that right at least, performed a series of chemical tests on cells from pus taken from discarded surgical bandages. Testing nuclei of the cells, Miescher found a phosphorus-containing substance that had an acid attached to it. In later studies, Miescher found a similar substance in salmon sperm cells. In doing these tests, Miescher had discovered deoxyribonucleic acid, or DNA again. Ugh, I just inhaled my own spit. After Miescher's discovery, an argument raged among scientists for nearly 100 years about whether DNA had the necessary chemical properties to code for both the cell's activities and its heredity. Later chemical tests showed that the DNA was a polymer containing 4 different subunits. These subunits were called bases, and given the letters A, adenine, G, guanine, C, cytosine, and T, thymine. Scientists could not imagine how only 4 different subunits could contain all the information needed to direct the cell's activities and heredity. It seems to them that the DNA polymer was too simple in its construction to be the key to cell activity and heredity. Most scientists expected the key molecule to be a protein, because proteins are much more complex molecules. A single protein is made up of a chain of unique combinations of up to 20 different amino acids. This chain then folds repeatedly in a specific pattern, forming the particular protein's characteristic shape. The protein's 3-dimensional shape is related to its function. Many scientists dedicated their research to searching for the protein responsible for cell activity and heredity, but a series of experiments begun in 1928 by Frederick Griffith eventually led to the discovery of the 3-dimensional structure and the functions of DNA. The body of work described below provided the evidence scientists needed to identify DNA as the key molecule for cell function and heredity. The work of Frederick Griffith. Frederick Griffith, a much easier name to pronounce than Miescher. Anyways, Frederick Griffith was a British medical officer. He was concerned about pneumonia, a disease of the lungs caused by a bacterium. In order to try to understand how the bacterium caused the disease, Griffith grew or cultured different variations or strains of the same pneumonia bacterium in his laboratory and studied their effects on mice. He grew a strain of disease-causing or pathogenic bacteria and another strain of harmless bacteria. When he injected mice with the pathogenic bacteria, they all developed pneumonia and died. When he injected mice with a harmless bacteria, they did not become sick, and they all lived. He wondered if he could kill the pathogenic bacteria by heating it. To test this idea, he heated the pathogenic bacteria, and after it cooled, he injected it into mice. The mice did not develop pneumonia, although it's kind of brutal, to be honest. These pictures summarize Griffith's experiment. Mice injected with pathogenic bacteria die. Mice injected with harmless bacteria live. Mice injected with pathogenic bacteria that have been heated do not die, they live. But when he mixed some of the heated or dead pathogenic bacteria with a living harmless strain of bacteria, the mice did develop pneumonia and they died. The picture below summarizes this portion of Griffith's experiment. Mice injected with harmless bacteria mixed with pathogenic bacteria that had been heated die. After repeated trials, all with the same results, Griffith concluded that the heat-killed pathogenic bacteria had somehow transformed into harmless bacteria into the pathogenic type. Again, after repeated trials, all with the same results, Griffith concluded that the heat-killed or dead pathogenic bacteria had somehow transformed the harmless bacteria into the pathogenic type. When he studied the next generation of this newly transformed bacteria, he found that it, too, was pathogenic. He had taken a harmless living bacterium, mixed it with a dead disease-causing bacterium, and caused a new transformed disease-causing bacterium that, when it reproduced, passed on its disease-causing capabilities to its offspring. The harmless bacteria became pathogenic. It was transformed. Griffith did not know how the dead pathogenic cells had transformed the harmless cells and their offspring into killers. Rawr. But he hypothesized that some particular chemical in the dead cells was somehow responsible for the transformation of the harmless cells. It would take many years, and the work of many people before scientists would understand what happened in Griffith's lab, including myself, because I have no idea what's going on, to be honest. Well, living that kind of life. The collaboration of Avery, Maslod, and McCarty. About 16 years after Griffith's work, Oswald Avery, Colin MacLeod, and all the C's. Macklin McCarty performed a set of important experiments that provided evidence that DNA, not protein, was indeed the molecule that coded for the cell's activities and its heredity. Avery's group repeated Griffith's experiments and searched for the mystery transforming agent. They purified various chemicals from the heat-killed pathogenic bacteria and mixed them with living harmless bacteria cells. The harmless bacteria became pathogenic. It was transformed only when they mixed with purified DNA from the dead pathogenic cells. No other chemical succeeded in transforming the harmless bacteria into a pathogen. Avery, Maslod, and McCarty announced their discovery in 1944, but many scientists still doubted that DNA could really be the transforming agent that was passed on to the next generation. They still thought DNA was too simple a molecule. More evidence was needed to convince the scientific world that DNA was indeed the key molecule for cell function and heredity. The Hershey-Chase experiment. In 1952, Alfred Hershey and Martha Chase conducted an experiment to confirm the role of DNA in heredity. They were doing experiments with viruses, which are very simple, consisting of only a small strand of DNA or RNA covered by a protective protein coating. Hershey and Chase used radioactive materials to mark or label different parts of the virus. In one experiment, Hershey and Chase labeled the protein coating on the outside of the virus with radioactive sulfur. Side note, sulfur forms part of the protein coating that holds the virus together. Viruses grown in bacteria, cells on plates containing radioactive sulfur use the radioactive sulfur in the protein coating. In the second experiment, Hershey and Chase labeled the DNA molecules inside the virus with a radioactive phosphorus. Phosphorus formed part of the DNA molecule. Virus grows in bacteria cells and plates containing radioactive phosphorus use different radioactive phosphorus in the DNA. Viruses reproduce by attacking a host cell. Again, in bold, viruses reproduce by attacking a host cell. Viruses reproduce by attacking a host cell, in this case a bacterium, and causing the host cell to use its own machinery and raw materials to make copies of the virus. Kind of like a zombie thing. The host cell makes thousands of copies of the virus, neglecting its own needs and filling up with virus copies until it can't hold anymore. At this point, the cell bursts, releasing thousands of viruses, each of which will attack surrounding cells. In this manner, the copies of a single virus can quickly infect many thousands of cells. Hershey and Chase used the sulfur-labeled virus and the phosphate-labeled virus to infect the same kind of bacteria, grown in two separate containers. After the bacteria in which container were killed by the virus, Hershey and Chase examined the location of the radioactive sulfur and phosphorus atoms. The diagram at right summarizes what the experiment showed. Once the bacterial cells were infected with the labeled viruses, the cells were put into a test tube and the centrifuge spun in order to spin off the viral pieces that were outside of the cells. The heavier bacteria cells formed a pellet on the bottom of the test tube. The lighter viruses floated on the top of the tube. Radioactive sulfur was found mostly in the top, virus layer of the tube. Radioactive phosphorus was found mostly in the bacteria layer. This meant that the phosphorus-labeled DNA was inside the bacteria, causing the bacteria cells to make new viruses. The sulfur-labeled protein coatings were not inside the bacteria, so they could not be causing the bacteria cells to manufacture viruses and spread the infection. Hershey and Chase went on to discover that the new viruses manufactured by the bacteria in the experiment also contained radioactive phosphorus. They concluded that the original labeled viruses had injected their DNA into the bacteria cells, and that the bacteria cells had used this viral DNA to make new. Side note: DNA was widely accepted as the most likely candidate for directing cell activities and heredity. Anyways, back to the original sentence. They concluded that the original labeled viruses had injected their DNA into the bacteria cells, and that the bacteria cells had used this viral DNA to make new viruses. The labeled proteins from the original viruses did not appear in the bacterial cells or in the newly manufactured viruses. Hershey and Chase had provided strong evidence that DNA, not protein, was the hereditary material, at least for viruses. After the Hershey-Chase experiments, DNA was widely accepted the most likely candidate for directing cell activities and heredity. Still to be determined was the three-dimensional structure of DNA and how the molecule functioned. Chargaff, Franklin, Watson, and Crick and the structure of DNA. Remember the four different subunits of DNA: A, adenine, G, guanine, C, cytosine, and T, thymine. In 1950, Erwin Chargaff discovered that no matter what cells he studied, the percentages of the four different subunits remained relatively constant. The total amounts of DNA might vary, but the percentage of guanine equaled the percentage of cytosine, and the percentage of adenine equaled the percentage of thymine. Chargaff always included that guanine always paired with cytosine, and that adenine always paired together with thymine. These findings, called Chargaff's rules, proved to be crucial in determining the structure of DNA. Rosalind Franklin, working in the laboratory of Maurice Wilkins at King's College in London, oh, that's a cool name, provided another critical piece of evidence. Franklin used a process called X-ray crystallography to create a photo of the structure of the crystallized DNA. This photo was not an actual picture of the DNA molecule. It did not show the details of the molecule's three-dimensional structure, but it did provide clues in the form of mathematical distances between certain atoms in the molecule. DNA is a polymer that is constructed from smaller, repeating units. James Watson and Francis Crick, two scientists at Cambridge University, were very excited about the Rosalind Franklin's X-ray crystallography photo of DNA. Using mathematical formulae to determine the distances between the atoms in the molecule, they determined that it must be helical in shape. Or, for those of you who don't know, the little spiral staircase kind of shape. Using this knowledge and incorporating Chargaff's rules regarding the pairing of bases, they began to work on a three-dimensional model of the DNA molecule. Watson and Crick's first model was a very complex single helix, which they determined was wrong. They set to work building another model. Their second model, the now familiar double helix, was completed in 1953. This model astounded scientists at the time, with its logic and simplicity. Watson and Crick, along with Wilkins, shared the Nobel Prize in 1962. Unfortunately, Rosalind Franklin had died several years later at the age of 38 of cancer. Her important role in the development of the DNA model was not acknowledged until many years later. I'm sorry, Rosalind. You'll be remembered. The double-stranded model of DNA, as presented by Watson and Crick, is shown in an illustration to the right. Like other large molecules in cells, DNA is a polymer that is constructed from smaller repeating units. In the case of DNA, the building blocks are called nucleotides, and each nucleotide contains a base, A, T, G, or C, plus a sugar and phosphate group. There are several features about Watson and Crick's model that fit the experimental data. The base pairs were connected at the Chargaff data required: guanine, cytosine, and adenine, thymine. The percentages were equal between G and C and A and T. Looking at the double helical structure of the DNA molecule from one end, it looks like the X-ray photo taken by Franklin and Wilkins. The DNA molecule could serve as a template for further copies and thereby pass information about the cell from one generation to the next. In this DNA model, the bases of A, T, G, and C complement one another. That is, each base always pairs up with other specific base. These base pairs thus form a template. If the base sequence of one side of the helix is known, the other can be determined using Chargaff's rules. The information stored in these sequences can be copied from the template and passed on to the next generation when the cell reproduces. One piece of the information storage puzzle, i.e. how DNA could store information passed on during cell reproduction, was solved. The remaining problem, then, was how did DNA actually code for the production of all the molecules required for cell survival and heredity? The answer to that question is the topic of the next activity. Written by Carolyn Cisongradi. I don't even know what that means. A bachelor's in science, something. BS in chemistry and MS in neuroscientist. Currently teaches at Palo Alto High School. Julie Reyes is a high school science teacher in San Francisco. Wonderful. And I hope you enjoyed my little reading of this, because I might or may not do this in the future. I'm just wondering if you like me to. Anyways, for all you Northwoodies out there, tell me how you liked it, and if I should continue doing more readings, because, to be honest, this helps me study as well as you. Well, maybe more me, but... Anyways, see you next time!